Chapter 1, Section 2, Structure and Activities of Microbial Cells. You have to remember, all living things on this planet are based on the unit of a cell, okay? Doesn't matter. Prokaryotes, eukaryotes, bacteria, archaea, fungi, protozoa, parasites, plants, animals, whatever. It's either one or millions, or as in the case of us, trillions of cells. When we look at it, all cells have some things in common, okay? There's going to be some form of a cytoplasmic or a cellular membrane. That lipid bilayer separates outside from inside, inside outside. Keeps stuff in, keeps stuff out. Okay, it's phospholipid bilayer. Which types of phospholipids? Depends on which organism. Prokaryote, eukaryote, plant, animal, whatever. The cytoplasm is going to be there. Cytoplasm is going to be an aqueous mixture. Okay, it's not water. So sometimes you'll hear me say, we're trillions of bags of water. No, it's a water-based mixture of macromolecules, amino acids, sugars, the components, the precursors of amino acids, the precursors of lipids and things like that. Ribosomes and all sorts of nucleic acid molecules. Just way too much to sit there and try to list them all out. Ribosomes are also going to be there. All living things have ribosomes. As we're going to see in later chapters, the ribosomes for us, eukaryotes, is going to be different than the ribosomes for prokaryotes, the bacteria. There's still going to be the large subunit, the small subunit, but the components that make up the large and the small will be different. And there's going to be some form of cell wall. For microbes, the majority of them, yeah. Okay, they have to have a cell wall because an individual cell, they are at the mercy of their environment. Is an isotonic, a hypertonic, a hypotonic, water rushes in, water rushes out, cells expand and burst, cells collapse, cell dies. So they need to have some form of a wall, nice hard structure to maintain the shape. Now, we animals do not have this, okay? All of our cells are squishy, but we see this in other eukaryotes, plants, fungi, protists. Yes, they can and will have cell walls to help maintain shape. So when you go and you compare prokaryotic cell to a eukaryotic cell, keep in mind the artist did not draw these to scale. The average quote-unquote eukaryotic cell is going to be anywhere from 10 to 100 times bigger than a prokaryotic, the average prokaryotic cell. But notice, cytoplasmic membrane, cytoplasmid, ribosomes, many of the same terms are going to be there. When it comes to prokaryotes, the bacteria and the archaea, no organelles, okay? No organelles is the name of the game. But here's the problem with that. In my class, any of my classes, right here is the major difference between a prokaryote and eukaryotic cell. Because I can find eukaryotic cells that do not have an ER, that do not have a Golgi, do not have lysosomes, but they're pretty much going to have a nucleus. So in my mind, that's the deciding factor. Are you talking prokaryote or are you talking eukaryotes? If there's a nucleus or somewhere in the lineage of the cell there was a nucleus, because you got to remember, not all your cells in your body have a nucleus. Your red blood cells don't have a nucleus. But the lineage of the cells that make, the lineage of the cells in your bone marrow to make those red blood cells had a nucleus at some point in time. The organelles may or may not be there. Mitochondria may or not be there. Peroxisomes may or may not be there. Lysosomes, ER, Golgi may or may not. May or may not. The genome. Remember a genome? This is the way we discuss the cell's full set of genes. For us eukaryotes, DNA. Us eukaryotes, the higher eukaryotes, it's going to be a linear chromosomes within our nucleus. Some of the lower forms of eukaryotes, some of the protists and whatnot, are going to have a single circular, large circular, but we're not going to worry about those right now. For all intents and purposes, the eukaryotic eukaryotic genomes are going to be measured in the billions, B, of base pairs. Prokaryotes, archaea, the bacteria, a single circular chromosome. Single circular chromosome that's measured in the millions, M. So it's about 1,000 times smaller than our genomes. Got to remember from your intro cell and molecular, from your genetics courses, if you've already taken those, we have introns and exons. They don't. But what a lot of them have that not very many of us eukaryotes have are little accessory chromosomes known as plasmids. Plasmids are going to be measured in the thousands of base pairs. Plasmids are going to carry 1, 2, maybe 3, 4 at the most I've seen genes. Little extra something something that the prokaryotes, the bacteria, can pass back and forth. All living things are going to have some form of enzymes. The enzymes are not magical pixie dust. They act as catalysts. A reaction that would happen on its own, they just make it happen more efficiently under a better time frame. There's going to be transcription, translation in every organism other than viruses. We see DNA to RNA is transcription, RNA to protein is translation. DNA replication, there's going to be some form of polymerases that are going to copy DNA, whether it's linear chromosome, circular chromosome, or the plasmid DNA. When you look at microbes, all cells are going to have some form of a structure that is detailed within the genome. The blueprint on enzymes and proteins, the structural proteins, will determine the shape of the cell. There's going to be a metabolism. Things get taken in, broken down, rebuilt, waste products going out. There's going to be growth. 1 cell becomes 2, 2 becomes 4, 4 becomes 8. Just like you started off with 1 cell, you know, rounds and rounds of mitosis. You get the trillions sitting there watching this. Turns out microbes do their own thing, but they don't do mitosis because they don't have multiple chromosomes or a nucleus to worry about. So as we're going to see in later chapters, it's just binary fission. 1 becomes 2, 2 splits becomes 4. Even though they're relatively simple compared to us, they have gone through, these microbes have gone through evolution. The microbes we have now, the different species we have now, have not always been here. They may have been around here, been around a long time, but they aren't the original. There were things that came before. You know, the textbook also talks about some properties, you know, that might be in cells. Differentiation. Some microbes, some bacteria, their cells can differentiate. Okay? You take something like Staphylococcus aureus, Escherichia coli. The cell will always be staph. The cell will always be E. coli. But there are others, such as Bacillus subtilis, Bacillus anthracis. You have a vegetative living cell that can differentiate and become a cell that is, well, a spore, non-metabolically active, and then it just sits there. We'll talk more about that in the chapters later. That spore, under the right conditions, will break open, and out comes a new vegetative cell that's metabolically active, taking things in, making and breaking things, giving off waste products. Even though I've said in a previous lecture for this chapter that, you know, these microbes, it's every cell for themselves, there still is some crosstalk between cells of the same species and between bacterial cells of different species. They're giving off molecules, parts of their waste products. They're putting things out into their aqueous environment. Other cells will take up that as a signal. They'll respond to it. We'll talk more about this, what they call quorum signaling, later. Many of the microbes are going to be motile. They're going to have flagella or some form of cilium that allows them to move, go towards nutrient source, go away from bad things, something that could be bad. And then they have what they would call horizontal gene transfer, those plasmids, past the plasmids, back and forth. Maybe that plasmid has a gene, for penicillinase, the enzyme that breaks penicillin down that allows bacteria to be penicillin resistant. Or maybe it's the cholera toxin, which is why, E. coli, can be deadly. It picked up a plasmid that had either the cholera toxin or the Shiga toxin from Shigella, and now you have bloody diarrhea because you ate a Jack in the Box. And we'll talk more about that in a different lecture. So this motility, differentiation, cellular communication, these are all things that some bacteria, some microbes can do, but not all. And it's going to be how we separate things out. Because remember, as biologists, we like to pigeonhole things, group like with like. And, well, some of the criteria we use. Are they motile? Yes, no. Do they differentiate by forming a spore or some other type of fruiting body? What quorum signals, what intercellular communications do they use? Things like that.